few definitions there. Most of these are revision from year 10. So the allele is the different type of the specific gene. So for example, like with hair color, black hair, blonde hair, red hair, they're all alleles. Understand what I mean when I have the S dot, just means singular. So singular is locus. So just like, if we were to compare, to help me understand exactly what you're asking, what you're saying is, like, would your stomach be more efficient potentially than my stomach? Is that what you mean?
Yeah, the way.
Because it's different phenotype?
Yeah.
Which would be caused by different genes.
And what is the allele, like the combination of the phenotype? Or the gene
The allele is the way that we try to link the gene with the phenotype. It's the way that we describe what phenotype a specific sequence codes for.
So it's like the gene chooses an allele which then determines the phenotype. So like allele is like a set of options which the gene could code for.
Yeah.
Yeah.
And then whatever it codes for, depending on which is dominant, which is recessive, ends up being the phenotype. Yeah.
Would you say the colour of your skin is an allele?
You would say the colour of your skin is a phenotype caused by a particular allele for skin colour. So you're getting, it's really the language I'm trying to describe. Maybe the table's not particularly helpful. It helps me, but I mean, it might not help you.
We can just need phenotype on the other side of the allele.
Form of a gene with a specific phenotype.
how the gene affects the phenotype.
Yes.
Okay.
Isn't a phenotype like an end result because the gene has chosen a specific, well, not chosen, but
Yeah.
has gone through a
So,
specific.
yes, but so the allele is the link between the gene and the trait. It's just our word though. All I'm trying to so this is just a language then. It's just saying, okay, if we're talking about, if you're asked a question, explain the allele for the trait hair color, you would talk about brown allele, black allele, blonde allele, red allele. Because it's the link between them. But the way that we talk about it when we're talking about inheritance and all that sort of thing is we consider it as part of the genetic structure. So it's like a go-between between each. It's the gene that makes a specific phenotype. Sequence of DNA is probably a better term even than gene. Sequence of DNA makes a specific phenotype. That's not really a definition, that's just trying to use it in language. If that's not helpful, leave it alone, that's fine. That's just trying to explain. We then obviously come to, so that's diploid cells, where we have both copies, where we have both homologs, both copies of each chromosome. In haploid cells, sex cells in us, gametes, sex cells formed in meiosis, essentially just two mitotic events occurring concurrently. As DNA replication only occurs at the start of meiosis, where the chromosomes are split twice, so anaphase 1 to split homolog pairs, anaphase 2 to split sister chromatids, meiosis results in haploid cells. Cells with half the number, or half the diploid number, of chromosomes. We give the haploid number a symbol, we call it N, or often it's italicized, so it'll be italics N, and that the haploid number for an organism is just half the diploid number. It's often used to describe the number of chromosomes in gametes, this symbol N. As diploid cells have double the number of chromosomes of haploid cells, they are often given the symbol 2N, or 2 times N, because they have double the haploid number. Now obviously, because we are diploid organisms, haploid cells are not very useful in repairing our body. We don't make new cells for our skin or our muscles or our organs using meiosis. We use mitosis so that we end up with diploid cells. These are not cells that are going to divide unless they are fertilized. Well, at least in the case of eggs, or unless they do the fertilizing in the case of sperm. So these are a unique group of cells. Come about because of or through meiosis, which basically is two mitotic events. So we have interphase, cell duplication of chromosomes, or duplication of DNA. We have prophase, metaphase, anaphase, telophase. Then we don't have interphase again. We go from telophase straight to prophase, metaphase, anaphase, and telophase again. Then we have cytokinesis and 4 cells form. Because we've got 2 rounds of mitosis, we end up with 4 cells in total. And because we have 2 rounds of anaphase without a second round of division or a second round of duplication of DNA, we end up with half the total chromosome number. Half the number of DNA, or half the amount of DNA to what we started with, as well as half the number of chromosomes. We haven't talked yet about independent assortment or crossing over, and we will do in a moment. Has everybody got that table down? Those who want it. go through the steps to do with meiosis. Interphase is the same. Now, we don't talk about G1, S, or G2, not necessarily because they don't exist, but because it's a slightly more complex process. In females, the cells are actually paused at a particular stage of meiosis at birth, before they're allowed to continue. In males, they start, or they're sort of stalled at an odd stage of interphase. So we don't consider interphase in the same level of detail that we did for mitosis for meiosis. We just basically say the chromosomes replicate. So we have DNA replication. We then go into prophase. So prophase, the cells condense, the chromosomes condense, the nucleus dissolves. As the nucleus dissolves, the chromosomes are then free to start to move towards the metaphase plate. So we're entering metaphase now. Once they're aligned on the metaphase plate, and once they're aligned on the equator, spindle fibers form, and they pull apart the homologous pairs. So unlike mitosis, where each chromosome lines one on top of the other, during meiosis, the chromosomes line up with their pairs. So they're standing like receptions, holding hands. And the spindle fibers pull the chromosome pairs apart, so they separate the chromosome pairs into what are now haploid cells. Haploid cells, but with double the amount of DNA still. They still have those duplicated chromosomes. Now we go through prophase again. The nucleus doesn't necessarily reform. I don't exactly remember whether it does or it doesn't, and you don't particularly need to. But we go through prophase. Metaphase, this chromosome starts to line up along the equator again. Anaphase, this time because there are no homologs, the spindle fibers attach to each side of the sister chromatids. They pull the sister chromatids apart, separating them, and we now have or at the end of telophase and cytokinesis, we have our 4 genetically unique cells. Now, a couple of steps there are critical. During metaphase, or leading up to metaphase 1, during prophase 1 leading up to metaphase 1, there is nothing controlling which side of the cell each chromosome pair ends up on. Now, if you only look at one instance of meiosis, then that doesn't seem particularly important. But when you consider that conservative estimates are that girls at the start of their reproductive life have about 1 million viable eggs, and men produce about 100 to 200 million viable sperm with each ejaculation, that chance becomes incredibly important. For one individual cell, the likelihood that it is going to be genetically identical just from independent assortment to any other cell in the gametes, or in the gonads, is 1 in 23 million. No, 1 in 8 million, When you then factor in the next step, crossing over, that factor becomes 1 in 200 to 1 in 300 million. So the amount of variation we can generate in meiosis is terrifically large. Does that make sense? So I know I'm skipping over this reasonably quickly, but it's because we've covered it before. So does independent assortment, is that something you're confident in? Because we might as well spend a bit more time with it if you're not. But science is golden, so I'll carry on if not. We'll talk a bit more about crossing over. So all stages of sexual reproduction are energy intensive. We've already talked about that. So the organism has to achieve or has to receive some significant benefit from actually going through all of this trouble. The benefit is genetic variation. The genetic variability that is seen in sexual reproduction is just tremendously large. Sexual reproducers have these 2 unique mechanisms, independent assortment and crossing over. What is the mechanism for variation that's shared among sexual and asexual reproducers? Mutations. So there is that 3rd form of variation. I'll get rid of these because it's easier. Everybody's got these definitions. So different working with year 12s who can write at a good pace. Being recorded now, so I'm not going to make the distinction any further than that. But it is nice. Should actually put genetic variation. crossing over is interesting. Crossing over is what happens, or is what results in some really unusual variations in offspring. Genes are linked. So, for example, the gene for eye colour is often linked to hair colour. Blonde hair goes with blue eyes. Light skin goes with light hair colour. Those are genes that are linked. Every now and then something odd happens. So, for example, those genes that are linked, perhaps you've got a father who has dark hair and dark eyes. Let's say dark brown and brown eyes. So dark brown hair and brown eyes. But he inherited a recessive trait from his mother for blonde hair and blue eyes. And during meiosis, the dark hair, dark eyes gene, or the dark hair, dark eyes chromosome, switched eye colour. So that instead of having dark hair, dark eyes, we had dark hair and light coloured eyes. So now we have dark brown hair with blue eyes. That cell, that sperm cell, goes on with dark hair and light coloured eyes and blue eyes, goes on to fertilise an egg. And that child ends up with dark coloured hair and blue eyes. Would be the first instance of that in that particular family. So crossing over can break traditional, like I suppose you could say, like family linkages. Really, really interesting little section of variation there. And it throws the spanner in the works every now and then. Sometimes really unusual things happen with crossing over, but that's the whole point of it. Weird things are supposed to happen. That's the point of variation. That's the way this whole process has been designed. So that unusual things can happen, because perhaps that's useful. So where we've got crossing over. Crossing over occurs in prophase 1. I should say metaphase 1. Can't happen in prophase 1. So it happens in metaphase 1 when homologous pairs line up on the metaphase plate. Better fix that. So the homologs are joined at various loci, and these loci are called chiasmata. Chiasma. Does anybody know what a chiasma is, or a chimera? Pardon? Yeah, it's a special kind of hybrid though. It's a messed up kind of hybrid, is a chimera. Of a teen wolf.
Yeah.
Yeah. Pardon? Full metal? Very good.
Yeah.
So, in mythology, a chimera is a mix between different types of animals. So, like in Greek mythology, a chimera is often a mix between a dragon and a lion or something like that. It sort of varies. Chiasmata have a similar sort of nature. A chiasma is a joining, I suppose, an intermix or an intermingling. At this stage, the homologs are said to be bivalent. And I'll show you why. The chromatids are now referred to as a tetrad, because there are no longer 2 of them, there are 4 of them. At this stage, endonucleases, so special kinds of enzymes. What's a nuclease? Is an enzyme that? Yeah. An endonuclease is a special kind of nuclease that is able to snip genes at specific locations. Endo meaning within. It also can mean preserving. So this is like a preserving kind of nuclease. I suppose what I'm trying to get at there is this isn't going to degrade the whole chromosome. This is only going to degrade specific sections or cut out specific sections. Yes, I think so. Yeah, so endonucleases, these specific enzymes, they snip the innermost non-sister chromatids at these chiasma, and then the sections of DNA swap. The process is called crossing over and produces new combinations of linked genes. Before I get these definitions down, this will be the last thing we do today. So we've got that's just independent of solvent. That's pretty simple. This will be the last thing we do today, and we'll have 12 slides to get through tomorrow, which should be pretty easy. So, we've got our chromosomes. Kind of need to. I might actually blow this up a bit bigger and draw it a bit more carefully. It's a bit dramatized, but hopefully it'll make things a bit clearer. So then there are these special linkages that are formed. So these chiasma and the endonucleases come along and they snip the genetic information. Won't necessarily snip it at all chiasma. Well, they might. And that then goes on to produce a really interesting kind of chromosome. So the outermost sides, they're preserved. Those inner sections are now quite different. So we've switched those regions. And it doesn't matter which region you draw as switching, the key is that they switch. No genetic material is lost, that would be a disaster. So those chiasma form physical links that cause or that allow that swapping over. Now I don't know the exact biochemical nature of them. I don't know exactly how they do that, but they do.
Do you enjoy that free 0.1 second you get when you write at like that?
I do. Really do.
Because it's so hard to just add the T.
Yes.
What is tetra?
Okay, so tetra is 4. So this is joined together. The chiasma join the two homologous pairs. So the reason it's called a tetrad is because it has 1, 2, 3, 4 chromatids. It's bivalent because we now have 2 opposing sides. We've got the sides that are away from or that are opposing each other, I suppose, or far, and then we have the inner sides.
Is there a book called Iron 4?
Is there a
Book called Iron 4?
Yeah.
No, how Iron, yeah, wouldn't it be cool if it was like Iron Tetra? Like, that sounds so much sick.
It does sound so much cooler.
It does. So much sick. So much sick, bro. Shouldn't be saying that. Let's try.
actually sure if endonucleases are only involved in crossing over, but it's certainly key. What are some examples of linked genes?